Dear students, in this lecture, I am going to describe the different forms of DNA. In previous lecture, I have already explained the structure of DNA. So now the different forms of DNA. As you know that DNA stands for deoxyribonucleic acid, which is genetic material, and it is found in nucleus. Small amount of DNA is also present in mitochondria in the eukaryotes. So you can see the structure of cell, then nucleus, and this is the helical structure of DNA. So DNA structure was given by Watson and Crick in 1953. So eukaryotic organisms such as animals, plants and fungi store the majority of their DNA inside the cell nucleus, and some of their DNA is organelle such as mitochondria. So based on the location of DNA it may be of two kinds. Nuclear DNA, here the DNA is located in the nucleus and usually has two copies per cell. So structure of nuclear DNA in case of human being, it is linear with open ends and includes the 46 chromosomes containing 3 billion nucleotides. So nuclear DNA is diploid, ordinarily inheriting the DNA from two parents. The mutation rate for nuclear DNA is less than 0.3%. The mitochondrial DNA, it is located in the mitochondria and contains about 100 to 1000 copies per cell. So mitochondrial DNA chromosomes usually have closed circular structures and contain, for example, 16,569 nucleotides in human being. So mitochondrial DNA is haploid, coming only from the mother. So mutation rate for mitochondrial DNA is generally higher than the nuclear DNA. Then you already know the structure of DNA, that it is a double-stranded helix, and each DNA molecule is composed of two biopolymer strands coiling around each other to form a double helix structure. These two DNA strands are called polynucleotides as they are made of simpler monomer units called the nucleotides. So this is the B structure of DNA, B type. Here you can see the double helix structure and it is taking the turns, and in each turn there are 10 nucleotide base pairs in case of bDNA. There may be the variation in the base pairs in other DNA, other types of DNA. So components of nucleotides, you already know that it composed of the nitrogenous base, ribose sugar and the phosphate group. So nitrogenous base may be pyrimidines and purines. So pyrimidines are generally cytosine, thymine and uracil, which is single ring compound. And purines are double ring compound such as adenine and guanine. So now the forms of DNA. So there are different forms of DNA depending upon many factors such as salt concentration, DNA sequence, quantity and direction of supercoiling, modified bases, metal ions and presence of polyamines. So mainly DNA is the bDNA or B form of DNA, which I have already described to you. So in certain conditions, different forms of DNA are found to be appear like aDNA, zDNA, cDNA, dDNA and eDNA. So there are different forms of DNA. So why there are different forms of DNA? As simply there is not enough room for the DNA to be stretched out in a perfect linear bDNA conformation. So in nearly all the cells, from simple bacteria through complex eukaryotes, the DNA must be compacted by more than 1,000 folds in order to even fit inside the cell or the nucleus. So different forms of DNA. First, I'm going to give the structure of B form of helix, which is already described, and it was given by Watson and Crick, and it is mostly the most of the DNA is of B form of DNA. It is right-handed helix, and there are the turns, and every turn takes place at 3.4 nanometer. Each turn contains the 10 nucleotide base pairs, and it contains the two grooves, major grooves and minor grooves. So this is the B form of DNA. You can see the structure of B form of DNA showing the major grooves and minor grooves. So bDNA is a Watson and Crick model of double helix. So already know about the bDNA. So now, as in the bDNA, there is double helical model if pyrimidine of one strand is always paired with purine on the other. So from Chargaff's rule, the two strands will pair adenine with thymine and guanine with cytosine. So this pairs a keto base with an amino base, a purine with a pyrimidine. So two hydrogen bonds can be formed between adenine and thymine, and three can be formed between guanine and cytosine. So these are the complementary base pairs. So base pairing scheme immediately suggests a way to replicate and copy the genetic information. Now the A form of DNA. It is less common form of DNA and more common in RNA. It is right-handed helix, and there are 11 base pairs per turn with a 0.26 nanometer axial rise, and the 28 angstrom 28-degree helix pitch, and it contains two different grooves, major grooves which is very deep and narrow, minor groove very shallow and wide, which is a binding site for the RNA. So this is the diagram showing the A form of DNA. The major difference between A form and B form nucleic acid is in the conformation of the deoxyribose sugar ring. It is the C2 endo conformation for B form, whereas it is a C3 endo conformation in the A form. So a second major difference between A form and B form of nucleic acid is the placement of base pairs within the duplex. So in B form, the base pairs are almost centered over the helical axis, but in A form, they are displaced away from the central axis and closer to the major groove. The result is a ribbon-like helix with the more open cylindrical core in the A form. Now the Z form. So there is radical change of B form and it forms the Z form. It is left-handed helix, very extended, and it is rich in guanine and cytosine. The sugar base backbone form the zigzag shape. So Z-DNA can form when the DNA is an alternating purine pyrimidine sequences such as GC, 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 and indeed the G and C nucleotides are in different conformations. The B to Z transition of DNA molecule may play a role in gene regulations. So it has antiparallel strands as bDNA. It is long and thin as compared to the bDNA. And there are 12 base pairs per turn and twist angle 13 and helix diameter is 1.9 nanometer. So big difference is at the G nucleotide. It has the sugar in the C3 endo conformation like A form and in contrast to the B form. And guanine base is in the syn conformation. This places a guanine back over the sugar ring in contrast to the usual anti-conformation seen in the A and B form of nucleic acid. So this is the structure and showing the two mononucleotides units of bDNA. Here you can see this is the fifth position, this is the fifth position, this is the third position. Now this is the dinucleotide unit of Z-DNA. This is the position 5th, and this is second one is the position of the 5th. This is the third. So adjacent sugar residues have alternating orienting repeating units is a dinucleotide. So this is a Z-DNA. So now you can see the different forms of DNA. This one, A-DNA, B-DNA, and Z-DNA. This is a zigzag form. So A-DNA twist 33, B-DNA twist 36, and C to Z-DNA 30. And you can see the difference here, which is given in the tabulated form. So these are the main differences between A-DNA, B-DNA, and C to Z-DNA. So other forms of DNA like cDNA, it is formed at 66% relative humidity in the presence of metal ions. It is right-handed with the axial rise of 3.32 per base pair, and there are 33 base pairs per turn, and it has a diameter of 19 angstrom and the tilt base is 7.8. This is the cDNA. Then ddNA, it is a rare variant with 8 base pairs per helical turn. So these forms of DNA found in some DNA molecule devoid of guanine. eDNA, it is extended DNA and it has long helical axis rise and base perpendicular to the helical axis. So conditions favoring A form, B form and Z form of DNA. Whether a DNA sequence will be in the A, B or Z DNA conformation depends on at least 3 conditions. The first is the ionic or hydration environment, which can facilitate conversion between different helical forms. A DNA is favored by low hydration, whereas Z DNA can be favored by high salt. The second condition is the DNA sequence. A DNA is favored by certain stretches of purines and pyrimidines, whereas Z DNA can be most readily formed by alternating purines and pyrimidine bases. The third condition is the presence of proteins that can bind to DNA in one helical conformation and force the DNA to adopt a different conformation, such as proteins which bind to bDNA can drive it to either A or Z forms. In living cells, most of the DNA is in a mixture of A and B DNA conformations, with a very few small regions capable of forming the Z DNA. So then there are the terms denaturing and annealing of DNA. The DNA double strands can be denatured if heated or treated with chemicals. DNA denaturation is a reversible process as denatured strands can re-annealed again if cooled. This process can be monitored using the hyperchromicity or melting profile. So hyperchromicity or melting profile, it is used to monitor the DNA denaturation and annealing. It is based on the fact that single-stranded DNA gives higher absorption reading than the double-stranded at wavelength 260. Using melting profile, we can differentiate between single-stranded and double-stranded DNA. So this is all about the different forms of DNA. So most common and stable form of DNA is the bDNA and zigzag form is the ZDNA. And there are other forms such as A form, D form and C form. So this is a structure of ADNA, bDNA and ZDNA. Thanks.